I'd like to give you an overview of DILIsym, which is a mechanistic mathematical model of drug-induced liver injury. By way of background, DILIsym was developed with the support of the DILIsym initiative. The DILIsym initiative has included as many as 16 major pharmaceutical companies who have provided not only financial but intellectual support in developing the qualitative and quantitative and software capabilities that are now included within the DILIsym software. DILIsym is, as a mathematical model, we'd like to describe it as middle out and multi-scale. There are various approaches to mathematical modeling of physiological systems, some being described as the bottom up, some as top down. Really with DILIsym, by virtue of the fact that we're focused on the organ, the liver, and the tissues within, we really focus on the middle of this, hence the term middle out. That said, depending on the type of biochemistry representing, there are some aspects, some areas of DILIsym where we really drill down and get into kind of a bottom-up sort of very biochemical representation, such as the diagram for mitochondrial dysfunction describes. We also kind of scale back out and include multiple systems, particularly when we account for drug distribution and metabolism, and interpatient variability. So again, it is multi-scale, capturing some elements of biochemistry and molecular biology, with the physiology and the intertissue distribution and uptake of drugs, cellular processes, all the way to interpatient variability. DILIsym represents 4 species: the human, the rat, the mouse, and the dog. So that includes, of course, the primary preclinical species, as well as population variability within each of these species, particularly individual or interpatient variability. DILIsym also includes 3 zones within the hepatic acinus, and this enables us to be consistent with histology type measurements. We've really explored the mechanisms of DILIsym to a great extent, and DILIsym represents these mechanisms, and this summary diagram describes how these mechanisms interact. Each of these nodes represents a submodel within DILIsym, and how the various submodels interact. And if, for instance, if we focus on the upper left, you can see that the drug metabolism distribution provides predictions of the hepatic exposure of parent and or metabolite for a given compound. And then there's the potential to interact with bile acid homeostasis, lipotoxicity, generation of reactive metabolites, reactive oxygen species, and mitochondrial dysfunction, all of which have the capability of interacting and disturbing the hepatocyte life cycle, in other words, inducing apoptosis and or necrosis. There's also an interaction with a representation of the innate immune system, which can exacerbate injury or ameliorate injury. It also leads to provides support for the regeneration post-injury. We also have a submodel of various biomarkers of drug-induced liver injury, and we'll touch on some of these aspects later in this presentation. So the diagram really describes the qualitative interactions. In order to capture the quantitative aspects, we've relied on what we call exemplar drugs of hepatotoxicity, and you can see them listed on the right side of the slide here, where we've kind of grouped them into their mechanistic categories: reactive metabolites, mitochondrial dysfunction, and bile acid transporter inhibition. You can see that some compounds span multiple mechanisms. And again, we've used this to establish the quantitative interactions within and between the submodels captured in this summary diagram. With DILIsym, we can represent numerous dosing protocols, from multiple dose to single dose to overdose, intraperitoneal, oral, intravenous. A number of possibilities are all captured. DILIsym is what we like to describe as compartment-based modeling. It's a relatively large model with over 500 state variables. It's primarily ordinary differential equations, and it's built on the MATLAB platform. And we have developed a graphical user interface to enhance kind of the point-and-click usability of it. DILIsym also includes what we like to call a form-to-function connection, where the loss of hepatocytes due to injury comes back and affects the ensuing uptake of the drug and circulating metabolites to retain mass balance. So I'd like to expand a little bit on the use of the exemplar compounds. So again, the value proposition of DILIsym really lies in the fact that we can link an unknown or relatively new drug, i.e. candidate X, mechanistically to be comparable to these exemplar compounds. And so this is, the diagram more or less illustrates this. So we have acetaminophen, tolcapone, and bosentan as exemplar compounds, and the three categories listed on the left. And inasmuch as candidate X and has qualities that are comparable in oxidative stress, in mitochondrial toxicity, and in bile acid transporter inhibition, as these exemplar compounds, we can then predict injury based on the measurement of the features of that compound, the characteristics of that compound, and how it disturbs these processes. And again, it's not, I should also point out that these aren't necessarily separated. Multiple mechanisms can be active simultaneously within DILIsym, and again, many of these exemplar compounds that we've included as well interact with multiple mechanisms. So as far as data inputs, in order to establish what the appropriate parameter values are describing a particular new chemical entity, we have them in these categories that are listed up here. So kind of drug absorption, distribution, drug metabolism, and hepatotoxicity mechanisms, those first three on the top. As far as kind of the classic DMPK inputs go, really we're relying on classic data, physical chemical characteristics, protein binding, and the like. As far as the hepatotoxicity mechanisms, and really this is what separates DILIsym from other mathematical models, we have identified in kind of these key areas a set of inputs, in other words, in vitro assays that can be done that can describe then the characteristics of hepatotoxicity mechanisms for a given compound. And as far as outputs go, by and large, the DILI community relies on biomarkers of injury, and we have represented a substantial number of drug-induced liver injury biomarkers, and these are listed here in the table. And you can see we have kind of the classic measurements: ALT, bilirubin, AST, prothrombin time. All of those are included within DILIsym. And we've also included some of the more recent emerging biomarkers of DILI, such as HMGB1, cytokinin 18, both full-length and cleaved, SDH, arginase, and miR-122. And so these are all captured, like I say, are represented within DILIsym. We also have the ability to represent a number of other model outputs, and some really critical ones for understanding simulations are the fraction of viable hepatocytes, liver ATP, as well as many, the exposure within the liver, the circulation, etc. So various PK outputs as well. In addition to simulating injury, we also have the ability with DILIsym to simulate the regeneration or the post-injury proliferation rates. And we have been able to ascertain a variety of data sets, such as described by Portman 1975 in patients after acetaminophen overdose, in which liver biopsies were performed over the space of several weeks following injury. And you can see that the predicted recovery rates are consistent with what was measured in these patients. And again, we have a number of other data sets. I should point out that the simulation results shown in red really represent, in this case, just the baseline simulated human. We can capture a variability in this response by using what we call simulated populations, or SimPops. So the SimPops really are population samples with variability in the hepatotoxic drug responses, and that variability is due to variability in parameter values across a variety of different biochemical areas. And so really what we've done is we've gone into the literature and identified not only what kind of the classic mean value for a given parameter would be, as an example, baseline glutathione, but we also established what the literature defines as the range, the upper and lower limits, as well as the distribution of glutathione, for instance. And then we do the same for each of the various parameters, and use the combinatorics, combine them all into unique possible simulated patients. We run a protocol just to kind of validate, in this case, to validate that each of these simulated patients are still appropriate. In this case, we used data from Davis 1976, looking at bilirubin levels following acetaminophen overdose. And you can see that, for these three patients, that they seem to be consistent with the range of reported data in the Davis paper. And then we build up the number of patients within the SimPops, typically in the range of 100 to 300 patients, simulated patients, again, ensuring that they are within the range. And then once we have some confidence that we have aggregated a SimPops with viable simulated patients, we then are able to use that SimPops to make predictions with new chemical entities, what we're calling compound hex, and look at biomarkers, look at mechanistic underpinnings and the like. So we've been able to now, at this point, we've published a number of papers with DILIsym describing not only how it's put together, but also various applications, some of which are described here, kind of in vitro to in vivo extrapolation. We've been able to rank compounds by risk. We've aided in the study design for preclinical biomarkers, as well as dosing estimates in order to kind of establish the margin between safety and efficacy. We've also been able to predict variability in response, as well as interpreting clinical biomarkers to really understand the magnitude and nature of the injury. And there are many other possible applications of DILIsym as well, and we are excited about the possibility of using this mechanistic mathematical model of drug-induced liver injury going forward. Thank you for your time.